Okay, first thanks for the invitation to come and talk to you today and tell you a little bit about a project that we've been doing on rare diseases for the last 2.5 years. So this is part of a much larger project which is called Rare Disease Research UK, and it's funded by the Medical Research Council and NIHR. So I'm just going to give you an update on what we've been doing here in Swansea, telling you about new methods that we've got to diagnose rare diseases. So it's these are the Swansea team that's involved in this project. So Laura gave us a nice introduction to rare diseases, so I don't need to spend long on this. So rare disease is defined as one that affects less than 1 in 2,000 people. However, there are 7,000 known rare diseases. So this actually translates to a rare disease affects 1 in 17 people during their lifetimes. So they're individually rare, but actually collectively very common. And what is really quite disturbing is that only 5% of the rare diseases have actually an approved treatment for the disease. So this is a real problem in the rare disease space. So the project we're involved with, it's a collaboration between us in Swansea, our friends in Cardiff University and University Hospital Wales. So that's us down here. Then we've got UCL in London and St Mary's Hospital in Manchester, and we're all working together on this project. So the aim of the project is to actually just speed up the diagnosis of rare diseases using new technology called metabolomics and lipidomics methods that we currently only have in the research labs. So we've got these omics methods in our research labs, but they're yet to be in the NHS. And our goal is actually to use these methods to speed up the diagnosis of rare diseases, because diagnosis of rare diseases is a real problem, and often it takes up to 5 years for people to get a diagnosis. So once we've got these methods up and running, we want to then later translate them into the NHS metabolic labs, because in the metabolic labs they don't have these methods. So we've got the methods to diagnose, and also we want to use these methods so we can monitor the response to therapy. So the initial focus of our work is on lipid-related rare diseases. So we're really focusing in this particular project on rare diseases which are related to fat, really, because this happens to be our particular interest. So that's the project aims. So let's tell you a little bit about metabolites and lipids. So metabolites, they're the small molecules that are made in our body. So for examples of some metabolites are hormones, or they might be structural molecules which hold our cells together, or they might be breakdown products from the diet. So all these are metabolites. And actually our favourite metabolite in our lab is this one. This is cholesterol. So I've drawn the structure for those of you who have a biochemistry bent. Now cholesterol, it's a metabolite, but it's also a lipid or a fat. And an important point really of our work is that the patterns of metabolites in lipids in our body fluids, for example our urine or in our blood, change in response to disease. So if we have a rare disease, then the pattern of these metabolites in our blood is likely to be different from the normal population. So if we've got methods to measure these metabolites, then we can help in the diagnostic process. So we have this word omics. So omics really means measuring many things in one experiment. So we have metabolomics, and the idea of metabolomics is to measure all the metabolites that may be present in a biofluid, for example blood plasma or urine, or could even be a cell or a tissue. And then lipidomics, that's just a subsection of metabolomics. That's specifically looking at the lipids, or the fats. So lipidomics is the measurement of the lipid content of a biofluid cell or tissue. So the way that these omics techniques differ from conventional clinical chemistry lab work is in a conventional clinical chemistry lab, you'll just analyze 1 or 2 metabolites at a time. In using omics techniques, we'll analyze hundreds or even thousands of different molecules at a time. So this means that if you've got a disturbed pattern of molecules, there's more chance of us being able to observe that using these omics technologies. So that's the introduction. Here's just a slide, a protocol that we actually use. So we usually have blood plasma, sometimes we have urine, very occasionally we have cerebrospinal fluid from the spinal cord. We first extract the metabolites, so we throw away the proteins and the DNA, and then we separate the metabolites and we identify them using a method called LCMS. So first we separate the metabolites on this instrument, this LC thing, and then we identify and quantify the different metabolites using the mass spectrometer, the MS. So the output from the mass spectrometer looks something like this. We get a whole series of peaks, and the first approximation, each peak corresponds to a different metabolite. And the height of each peak, again as a first approximation, corresponds to the concentration of the metabolite. So we can get a list of the different metabolites in our blood plasma samples and the concentration of these different metabolites. And remember, people with a disease might have a different pattern of metabolites to the normal. So once we've got these patterns once we've got this list of metabolites and their quantities, we then go on to a metabolic pathway map. So these are maps which have been worked out over time of how different molecules in our body, one is converted into another. So we have these in the literature, and this is a metabolic map where each metabolite is in a blue square. And this shows us how different one metabolite is converted into another metabolite, and so on. So arrows show chemical reactions. Now if we have a rare disease where there might be a problem in one of the chemical reactions, let's say there's a problem in the chemical reaction down here in these arrows, what will happen then? Well, if we've got a problem in the pathway, we'll get a blockage in the pathway. If we have a blockage in the pathway, it's like a red light. So what happens if you've got a red light? Then the cars before the red light, the concentration of them builds, the amount of them builds up, so the metabolites build up. So these metabolites will build up. And we can measure these and we can identify them, so we can actually diagnose our rare disease. So by measuring the metabolites at the blockage, we can actually diagnose our rare disease. So that's what we're doing in our lab. So at the moment, we're diagnosing different rare diseases, and we're receiving samples from actually a large part of Wales in England. So we get the samples from Cardiff. So this really covers Wales, and then we get from the south, so from Southampton, lots of hospitals in London, from Cambridge, Birmingham, and then we got Manchester, Salford, Sheffield, Bradford, and Leeds. Yeah, that's our. So all these samples come to us to look at, and specifically they're coming to us to diagnose rare diseases. So sometimes most times it's for diagnosis. Sometimes it's actually to monitor experimental therapies. So there might be a rare disease and there's an experimental therapy, and then the samples will come to us, and we'll actually see if the therapy is working or not. So we're working in the rare disease space, so actually the samples are only coming to us when it's a rare disease that we actually have expertise for. So here are just a list of some of the common, some of the most popular rare diseases that actually come to us, and these are all lipid or fat related rare diseases. So these are rare diseases that our method works really well for. So this is diagnosis. We also have have therapies. So these are experimental therapies for this Wallman disease and this lathosterolysis, and we're actually monitoring these metabolites to see if we see a change in metabolic pattern, which can indicate that the treatment is working. And then we also look at some really ultra rare diseases where we're actually just trying to understand more about the biochemistry of these rare diseases. So there's this one, dihydrodolichol diphosphate synthase, which is incredibly rare. So there are actually only 6 families that show this disease. 6 in the whole UK, sorry. So the 6 in the whole UK, that translates to a prevalence of 1 in a million. So this is an incredibly rare disease. So unsurprisingly, little is known about the biochemistry of it. Now, very often, eventually, the clinician comes up with, when he's trying to diagnose a rare disease, he'll get a good idea what it is. So then he's got a good idea what it is, so he sends asks the patient to give a blood sample, and then they'll do a genomic test for that specific rare disease. But very often, in the rare disease space at least, you come across variants of uncertain significance. So a variant of uncertain significance is where there's a gene change that does not have enough evidence to show whether it causes a disease or not. So in other words, the genetic data is equivocal. And if the genetic data is equivocal, then the clinician does not have enough information to actually diagnose the disease. At this point, this is when the samples very often come to us, because we can actually provide extra evidence to confirm whether the disease is what the clinician suspects to be or not, as the case may be. So I've got a nice, I think it's a nice case study here of how we use this technology, and we're able to help in the diagnosis of a rare disease. So this rare disease I'm going to talk about here is called hereditary spastic paraplegia type 5A. So this starts as a walking disease, difficulty of walking in adolescence. So the adolescent has difficulty with walking, and later on they need a wheelchair. So it usually presents in adolescence, but sometimes it can present younger, sometimes it can present older. Now the genetics of the disease were discovered in 2008, and it knows we know that they're disease-causing mutations in a gene called CYP7B1, and this gene translates to a protein which is also called CYP7B1. Now this disorder, it's autosomal recessive, so that means that to have the disease, you need to inherit a defective gene from both your mother and your father. So these things run in families. And the prevalence of this particular disease is 1 in 100,000. So it's rather rare. Now it's known there are known to be about 600 different mutations in the CYP7B1 gene, and of that 600 mutations, about a third of them are variants of unknown significance. So when the clinician orders his genetic test, it's about a third, 1 in 3 chance that the genetics will come up with a variant of uncertain significance. So the clinician, to make a diagnosis, he then needs to order a biochemical test, and there's no biochemical test available on the NHS for SPG5 for this disorder. So this is when the samples come to us. They only come to us when there's no test on the NHS. So the samples come to us, we put it through our protocol, and this defective gene codes for a protein, CYP7B1, and that's an enzyme in the metabolic pathway of cholesterol. So that the enzyme doesn't work. So that means the substrates of the enzyme, these molecules above it, build up in concentration, and the concentration of the product of the enzyme, these molecules below my red line, these fall down. So this is something that we can easily measure with our technology. This is just for the biochemists amongst us. So the defective enzyme is this one, CYP7B1. It's appearing twice in this pathway, here and here. So the substrates of that enzyme, which are above it, the concentrations of this molecule, 26HC, and this molecule, 3-beta-HC. If the patient's got the disease, the concentration of these molecules should go up. So let's now look at some data. First, show you what the readout looks in a healthy sample. So we see this 26HC and this 3-beta-HCA. They're giving little peaks. We then take a blood sample from our suspected SPG5 patient, and whoop, up shoots the 26HC peak, up shoots the 3-beta-HCA peak, and diagnosis is just like that. So this is a really lovely example where we can make a diagnosis of this disorder. So this SPG5A, and we're finding more and more patients with this disorder, and we've actually diagnosed 5 patients in the last 6 months. So 3 of them came from London, 1 adult and two children, and both of them now are hopefully going to go on to some experimental therapy using statins, where hopefully the levels of these metabolites which have gone up will come down on statin treatment. Because it's suspected that these 2 metabolites could be toxic. We then got a patient quite recently from Cambridge, and this patient, the clinicians first suggested he first showed clinical indication of this type of disorder at the age of 40. But it's only now at the age of 80 that he's actually got a firm diagnosis. So you might think, well, that's not very important, the man's 80. But it's, of course, very important for his family, because this disorder is a genetic disorder, so it can run in the family. And finally, we just had a patient here diagnosed this week, which was from the Romford area. So here I've given you an idea of what we've been doing in our project. Of course, I've cherry-picked. I've only picked the examples where things work, and we do get many samples where we put them through our method, and we just don't see a change. So we're actually of no help at all. But that happens some of the times. But it also happens sometimes that we make a diagnosis, and this is a diagnosis that without our methods that just wouldn't have been made. So that's more or less it. Just to acknowledge the team. So this is our group, our team in Swansea. So we've got great collaborators, brilliant collaborators in Cardiff, UCL, Manchester, and Sheffield. And essentially, we're covering essentially all of Wales, because the patients will either go to Cardiff or they'll go to Manchester. We're also covering a large part of England, but actually at the moment we don't seem to see any rare disease samples from Scotland or Northern Ireland, but we'll try and correct that in the second part of this project. Okay, finally, thanks very much for listening.
Do we have any questions, please, for Bill? Alan.
There's a lot of questions common. I said earlier about cross-border collaboration and that. It's funny you said that Sheffield Children's Hospital.
Yes.
That's where my clinical consultant is based. I've also got SPG7 because they looked at SPG5.
Right.
So, and it was Claire.
Claire Hart is a lovely lady.
Yes.
So she's been incredibly helpful for us in this project. So
she's the one that suggested.
I will tell her.
So it does work.
Imogen, thank you.
How labour intensive is the protocol, and how easily would it be integrated into the NHS?
Very good question. So it is labour intensive, but it could be integrated in NHS. You can get a robot to do it, right? But robots are very expensive. and you've got to convince your healthcare commissioner to spend a lot of money on a robot for a rare disease, or rare diseases. So it's certainly very possible. Yeah.
Any further questions for Bill? Guy,
Guy.
I can see your hand.
Yeah, I was interested. What level of a metabolite would equal an iffy result, a result?
All right. So good question. Very good question. So with the easy ones, we normally see a 5-fold change, right?
Yeah.
So we see if it's a 5-fold change, then almost certainly we're pretty certain it's a diagnosis. But less than 5-fold, we're in the same position, the variants of uncertain significance.
Yeah.
Yeah.
Okay.
Yeah. So yeah.
And then what's the frequency of that happening in your tests?
That's a good. So we have the easy diagnosed, and that then falls into the undiagnosed. And I would maybe it's 50% of the samples that we get. But of course, people only send us the samples when they think we're going to get it.
Yeah.
So it's not that's not a fair comparison.
No. Okay.
But we have maybe a bit better than 50% success rate.
Good. Thank you.
Yeah, one more back. Thank you.
Thanks. So the people who already have genetic testing by the time you get their samples, would it be possible for a doctor to think, I think this might be a problem with lipids, and send you the samples when there's been no genetic testing, and then you can have a quick look and try and work with, you might...
Yeah. Of course, that would be possible, but I guess the route is genetics first. Yeah. Because of course genetics truly is high throughput, and we are very low throughput.
I've got a question over the top.
So the lipidomic diagnosis then, does that help upgrade the VUS status?
Right, exactly.
Does it feed into that system?
Yeah, exactly. So then you up. Things are never easy though. That should upgrade the VUS, right? But then of course we have to publish it. So we can tell you now what extra VUS there are, but first that has to be published. So there's this delay. Yeah.
Okay, I think you're off the hook now. Bill.
Well, thank you very much.
Thank you very much. Thank you very much.